Right, Heather,
All righty, I will take it away. Welcome everyone to the US BioGeoSCAPES Planning Workshop. My name is Heather Benway. I'm a senior research specialist here at the Woods Hole Oceanographic Institution and the executive officer of the Ocean Carbon and Biogeochemistry, or OCB project office, which is supported by NSF chemical and biological oceanography programs and the NASA Ocean Biology and Biogeochemistry program. OCB is a global network of scientists working across disciplines to understand the ocean's role in the carbon cycle and how marine ecosystems and biogeochemical cycles are responding to environmental change. The OCB office provides support to its network in many, many ways, including communication, science planning, education, capacity building, and sponsoring and coordinating community activities like these. I encourage you to visit our website or find us on social media to learn more about the breadth of OCB. You can see some of our major focus areas highlighted by the green icons on this slide. You can really see that the OCB works kind of at the nexus of climate, biogeochemistry, and marine ecosystems, marine life. I really want to emphasize here that OCB is a bottom-up program, meaning that the leadership and activities of OCB are shaped by its community through an open process. And as you'll see today throughout the presentations that are lined up, OCB has contributed to a decade-long history of oceanographic community championing to reach this point. And I'm really excited and looking forward to this conversation about how the US will contribute to BioGeoSCAPES. With that, I'd like to pass it off to workshop organizing committee member Naomi Levine to get us started. And I'm not sure anybody else is going to say it, so I want to say it now. I want to thank our organizing committee members who have put so much time and energy into putting this workshop together, including the OCB project office staff, May Mahegan and Mary Zawoski. We will have our organizing committee members presenting, so that I will let them introduce themselves when they present their slides. Thank you.
Thank you, Heather. And she obviously didn't include herself out loud in that, but she's done an amazing job in helping. Really critical to this organization, organizing this workshop. So thank you all for joining today. We had so hoped that we'd be able to meet in person, which is why we kept delaying it. But we are really happy to have over 154 researchers that registered. So a really large group of interests and from many different disciplines. And we're have worked really hard to try to make this an interactive discussion between all of the participants over the next 3 days. I wanted to start off, though we're in a virtual space, by acknowledging that this workshop was planned while working on ancestral lands of the Wabanaki, Tongva, Kumas, Siouan, Iroquois, and Algonquin nations from the organizing committee's home regions. And we honor and respect the many diverse Indigenous people still connected to these lands and the lands of our participants. We acknowledge the painful history of genocide and the substantial tradition and local knowledge that Indigenous people hold and sustain. So feel free to add your own, the Indigenous people from your own region to the chat if you'd like to. So I want to start off by just showing you the faces of the organizing committee that Heather mentioned briefly. We're just going to be the guides over this workshop. So Ben Twining, Max Sato, Alyson Santoro, Adrian Marchetti, Heather Benway, Mahegan, and definitely feel free to reach out to any of us if you have any questions, comments, or concerns over the workshop or after the workshop. And we're just here to kind of help facilitate the discussions. So I'm going to go through a quick overview of what to expect over the next 3 days. So today we're going to start off with an introduction, a brief history of BioGeoSCAPES, what the meeting goals are. If you have any questions during these talks, please add them in the chat, and we're going to have a Q&A session at the end of these introductory talks where we'll draw from the chat and have an open discussion with the whole group. We're then going to move into breakout discussions, focus around how to build BioGeoSCAPES, and we're going to do those discussions in Zoom in breakout rooms using Jamboards for note taking, and we'll give you more instructions right before the breakout session. After the breakout discussions, we'll all come back to plenary and then go off to a poster session in GatherTown, and we'll give you an introduction to GatherTown at that time. On day 2 is when we'll do the summary of the breakout rooms from day 1 so that we avoid taking up too much time today. And then we'll do a poster session in GatherTown. Day 2's discussion is going to focus on intercalibration. We'll get an update on that, and in addition to what BioGeoSCAPE hypotheses might look like. And then we'll end day 2 with some science talks selected from the participants. Day 3, we're going to start the day with a breakout discussion. Summary of the breakout discussion. We'll have some more science talks, another breakout talking about interdisciplinary science and how to move forward, and end with a view from NSF. So before we jump into the talks this morning, I wanted to just go over a code of conduct for this workshop. So it's really important that we allow everyone the opportunity to speak. This really is designed to be a workshop where we're bringing a really diverse group of people together, and we want to get everyone's thoughts and viewpoints. So please do not interrupt people who are speaking. Please speak with intention, noting what is relevant to the conversation, and be respectful to critique ideas and not individuals. And kind of in line with that, also to listen with intention. In addition, please try to attend to the well-being of the group and remain aware of others in the group. To remind you that OCB does not tolerate harassment, bullying, intimidation, or discrimination in any form, physical or verbal abuse of any kind, or deliberate disruption of presentations or any form of communication. So please be observant and be an ally. And definitely let any of the organizers know if there's any issues. So with that, logistics out of the way, I'm going to turn it over to Adrian, who's going to start us off.
Okay, thank you, Naomi. Are you guys seeing my slides okay here?
Yes.
Okay, great, thank you. So again, thank you very much for attending this this workshop. We all are looking very forward to your contributions and hopefully helping us get this program started somewhere down the line. Just to, my tasks here, I have 2 tasks basically for this presentation. Is to just introduce to you the concept of BioGeoSCAPES, as well as to give you a bit of a brief history of how we got to the point where we are here today. There are 4 talks lined up within this sort of introductory session. I'll be the first, followed by Alyson, and then Maite Maldonado will give a bit of an update on international efforts towards BioGeoSCAPES, and then finally Ben Twining will go into a bit more of what we envision are the hopefully the objectives of this workshop, and hopefully what will come out of it. So just to kind of give you a brief summary of what this program is about, I think an important component here is the tagline, essentially ocean metabolism and nutrient cycles on a changing planet. And I'm going to start from the back end here and then move forward in the sense that, in terms of the changing planet, I'm sure we're all aware of the events that are happening right now in Scotland in terms of the urgency to do something about climate change. And we know that our oceans are changing. And so part of the goal of this program is to actually be able to understand how the oceans and the ecosystems that are part of the oceans are changing along with climate change. In terms of nutrient cycles, we have a very sort of broad brush of what we envision to be included in nutrients, essentially everything from trace elements all the way up to complex compounds like vitamins, and everything in between, including things in certain forms of carbon that will also be included in that concept. And I want to spend a time just focusing on this ocean metabolism, because I think it's sort of important concept of the BioGeoSCAPES program, and that we can consider ocean metabolism as essentially the chemical processes that drive biological activities. And we can look at this in a number of different scales, whether it be at the sort of the subcellular scale and what's happening within the cell, working its way up to organisms and assemblages and the interactions between those organisms that constitute communities, all the way to sort of more looking at global scale ecosystem levels and how that is influencing nutrient cycling, biogeochemistry, and feedbacks. And with that, sort of, we've come up with a mission statement that essentially is derived from many sort of players that have been involved in this vision. And it's to basically improve our understanding of functioning and regulation of ocean metabolism and its interactions with nutrient cycling within the context of a hierarchical seascape perspective. Now, seascape may not be a term that everybody is familiar with. Essentially, it's taking that concept of a landscape and applying it to the ocean. But there's a very nice paper by Kavanaugh et al. That sort of goes through basically the reason why we should be using a concept of seascape when looking at monitoring, management, and conservation of, in particular, pelagic ecosystems. So I'd actually recommend that you do take a look at this paper if you haven't seen it already. And so now, just in terms of looking at a brief history of BioGeoSCAPES, there are many events that comprise this history. And I'd like to just upfront apologize if there are certain events that maybe should be on this list that are not. But essentially, this is sort of a timeline based on the collective memory of the organizing committee. But essentially, we're looking at meetings and workshops over the last decade, standardization and intercalibration efforts, conference sessions that have been held, complementary programs that have been conducted over the last few decades, and then community input and feedback, including this workshop today. So in just looking at this timeline, you can actually go back to 11 years ago when there was a workshop funded by OCB called Molecular Biology of Biogeochemistry that was held in Los Angeles that really sort of got the ball going in terms of trying to, again, bring together the chemical oceanographic community and those that were studying mainly sort of the omics approaches to biology. Coming out of that was a cruise that was basically called the Geomics Cruise. And just to provide a bit more information about this workshop, involved 60 participants. And essentially focused on, again, that use of molecular methods and linking it with ocean chemistry and biological activities. And out of that workshop came this Geomics Cruise. And this was a 1-week cruise that was basically along a section of Line P. And there were a number of publications that actually came about from that research effort. But most importantly, what I think it did is it identified issues and challenges to integrating chemical and biological measurements. And so we realized, especially from the omics side of the perspective, we had work to do. And that, it included things in terms of trying to figure out sampling protocols and processing, analysis of samples, bioinformatics, and then also how we actually are going to include standards and intercalibration between labs for these omics approaches. So with that, we then are moving forward to a workshop that was held at Lamont Doherty in 2016. And the participants of this workshop, essentially what it was looking at is we had the Geotraces product that was coming down the line, and it was trying to figure out ways in which we can actually use Geotraces for other applications. And that's where the concept of perhaps trying to see if we can link Geotraces with what was coming out of Tara Oceans came about. In addition to that, there was basically 2 years after a BioGeoSCAPES International Scoping meeting. And just to kind of go in a bit more detail there, again, from that initial Lamont Doherty workshop, we were really interested in saying, okay, can we actually figure out how we can combine Geotraces data with Tara Oceans? And at that time too, BioGeoTraces was starting, which included some biological measurements. But what was the challenge there is as we started to look at these two different global programs and try to sort of connect, say, Geotraces sampling with Tara, we realized that there was a spatial mismatch in that if you look at this map here, and this is just showing all the Tara sampling stations, there's a large sort of distance between where the nearest Geotraces station might be in the order of, and this is looking at degrees. So we're looking at hundreds to even thousands of kilometers distance between the two. And that doesn't even incorporate any of the temporal variation that could be occurring in the chemistry of the ocean, and again, also how that is influencing the biology. So from that, we ended up saying, well, what we're going to need to do here is probably see if we can envision a new way of, or a new program that can combine these sort of measurements. And so we had an international scoping meeting that was held in Woods Hole, and that was funded by sort of stitching together funding from SCORE, NSF, and the Moore Foundation. And it included 28 participants that it represented 10 different countries. And so these are the countries listed here, including the United Kingdom, US, Canada, Japan, China, France, Germany, Italy, Israel, and Australia. And from there, we really sort of decided that, or what we were trying to do is figure out how we can leverage the successes of these global omics surveys and trace metal programs into some sort of linked standardization framework. And so from there, BioGeoSCAPES was born. And so we came up with a name, we came up with a mission statement, and a plan to hopefully lead to the implementation of this program. From there, moving forward again, we have sort of now in 2019 and 2020, we had the initiation of these national planning meetings. So one of the outcomes of this meeting in Woods Hole was that these representatives of all these different countries would go back to their nations and try to figure out how their nation would contribute to a program like BioGeoSCAPES. Well, there have been meetings held in other countries, for reasons obvious to everybody. There's been delay in some, including the US, because of the pandemic, but we're here today and we're happy about that. And in addition to that, we've had, basically sessions at Ocean Sciences meetings that have been highlighting BioGeoSCAPES related research. And just wanted to put in a plug, we actually do have a session for the upcoming Ocean Sciences meeting in February. And moving forward again, we realized that intercalibration and standardization and intercomparison were very important. And so these activities now are ongoing. We had a workshop here at UNC in January of 2020, just before the pandemic really started. And Max Sato and the rest of the proteomics community have been making a lot of headway in terms of a metaproteomic intercomparison, and those efforts go back to 2018. So we'll be giving you updates about some of those activities that are ongoing. And tomorrow we have presentations by Paul Berube and Max Sato that will update us about those intercalibration efforts. And so this brings us to where we are now. And so we have our US BioGeoSCAPES planning meeting here. And so in the next few talks, we'll give you a bit more information about what we're hoping for the outcomes of that, including what are the next steps, which include an international BioGeoSCAPES meeting as we work towards the development of a science plan. And so with that, I will hand it over to Alyson.
Thanks, Adrian. So we'll take questions for all these introductory talks to, collectively at the end, but you can put them in the chat. Okay, my name's Alyson Santoro. I'm at the University of California, Santa Barbara, and I'm one of the organizers of the workshop. And my goals today are to give you an example of a few, not every, existing model for what a global investigation of ocean metabolism looks like, and then explore lessons learned from a successful international publicly funded oceanographic program, which is what we would like BioGeoSCAPES to be. And so the example of this first goal, I have two examples, the Global Ocean Sampling Expedition, or GOS, and the Terra Oceans Expedition, which you just heard a little bit about from Adrian, and we'll hear more about on Friday, probably from Daniele Iudicone. And as an example of this second point, I'd like to use the Geotraces program. So for the youngsters in the room that might not be familiar with the Global Ocean Sampling Expedition, the first phase of GOS ran from 2004 to 2006. And this expedition was a really pioneering metagenomic sampling effort. The research platform for this expedition was a sailing vessel. And because of that, there were some limitations to what they were able to accomplish in this first phase. It really focused on surface sampling. And it focused mostly on low latitude oceans, so tropics and subtropics. The expedition was coordinated by a single institution, the J. Craig Venter Institute. And again, because of the limitations of the sampling platform, there were limited biogeochemical measurements that were part of that program. The successor to GOS, at least in my mind, is clearly the Terra Oceans program, the first phase of which ran from 2009 to 2013. And Terra was also a largely metagenomic sampling campaign, but it vastly improved the sampling density at higher latitude oceans. It also improved the sampling resolution with depth. So Terra sampled 2 to 3 depths per station and included a really nice expansion of biological sampling into higher trophic levels, including zooplankton. The sampling and analysis for Terra was coordinated by a relatively small consortium and was funded almost exclusively with private funds. And again, this expedition was carried out on a sailing vessel, which limited their ability to sample the deep ocean, and also because of constraints on the laboratory space, limited, again, the biogeochemical measurements that could be made as part of this expedition. And this has required that investigations into metabolism and physiology of the microbes sampled by Terra, they've had to use some creative approaches in relating that to the distribution of particularly micronutrients in the ocean. So this is just one example that came out a couple of years ago in GBC, analyzing the microbial community level response to iron availability. And in this paper, they compared their metagenomic data to iron data retrieved from a global scale model of iron. And as Adrian showed in his talk, the proximity to actual measurements of dissolved iron in the ocean was not always as great as one might hope. So this modeling approach is only possible with a few elements. So it really highlights that having measurements of micronutrients or other parameters really genuinely co-located with measurements of microbial metabolism is really valuable. Another limitation in my mind that's near and dear to my heart is the limitation in depth resolution of these prior studies. And this is a figure that was published as part of a summary of the Spanish Malaspina expedition, which occurred in 2010. So they put together this really nice summary figure where the x-axis is the number of stations that were sampled as part of these programs, and the y-axis is the depth of sampling. So the star at the surface is there because if you put the number of samples that Terra sampled at the surface ocean, that's over 200. So it would be way off the scale of this plot. But you can see that for most depths in the mesopelagic, they were sampled fewer than 5 times. You can also see a huge sampling gap between 1,000 and 3,000 meters. And Malaspina did a relatively extensive job of sampling the deep bathypelagic at 4,000 meters depth. And so I want to emphasize that the 3 programs I've mentioned so far, GOS, Terra, and now just quickly mentioning Malaspina, those have been coordinated by single institutions or single consortia or single countries. And they really haven't had to deal with some of the same problems that a truly large-scale international program might have to deal with. I think one thing that was really shown in the Terra data set was the power of having samples that were collected with a single protocol processed in one way, which allowed a true intercomparison of samples across the ocean. But that's a lot easier to do when all the sampling occurs from one vessel and all the samples are processed in a single lab. So I wanted to turn to an example of an international program which tries to combine data from multiple platforms and multiple researchers. And that's the Geotraces program. So a very big thank you to Bob Anderson at Lamont and Mariko Hata at Hawaii for providing me with the following slides that I've adapted here. So Geotraces is an integrated geochemistry-driven program that investigates the distribution of trace metals in the ocean. I want to underline this statement of what Geotraces is, which is that it provides insights into processes that cannot be gained from the sampling of individual investigators. And I think we should keep that in mind this week. The mission statement of Geotraces is to identify processes and quantify fluxes that control the distributions of key trace elements and isotopes in the ocean and establish the sensitivity of these distributions to changing environmental conditions. So Geotraces is guided by a very concise statement of its mission, which is another thing we could keep in mind as we move through the next few days. It's an international effort. Even these numbers are a bit outdated at this point. There have been 96 cruises completed by 17 countries from all 6 inhabited continents. And behind any program is people. And these are the 6 geochemists that really kicked off the Geotraces program from 5 different countries. And these individuals organized a town hall at the 2001 Fall AGU. And that town hall meeting led to the first Open Science Workshop in France in 2003. And from that Open Science Workshop, the organizers submitted a proposal for a planning workshop to the United States National Science Foundation. This subsequently led to individual countries forming their own national groups, similar to what we're trying to do here for the United States and BioGeoSCAPES, and the development of a science plan. There were 12 different countries involved in the original science plan, which is now up to 38, and that was approved by the Scientific Committee on Oceanic Research in 2006. So compared to the timeline that Adrian just showed you, this might seem pretty rapid, but it's important to remember that the first US Geotraces cruise actually didn't happen until 2010, so nearly 10 years after that first town hall meeting. And I thought it would be interesting to just mention briefly how Geotraces first went about deciding where in the ocean they were going to go and exactly where they would sample. They organized this around basin planning meetings, one for the Pacific, one for the Atlantic, and one for the Indian Ocean. So before a section was decided on, it had to be championed by an individual person. The overall process, for those of you that aren't that familiar with Geotraces of how this works now for US scientists, a management proposal is submitted to the National Science Foundation. There's a planning workshop that occurs, and scientists can advocate for specifics of the cruise and express their interest in going. And then individual PIs submit proposals to NSF to be able to participate in the cruise. Normal cruise planning activities happen, post-cruise meetings happen, and the data that results from Geotraces cruises, and I think this is a really important consideration for any new program, is what's going to happen to the data. For Geotraces, those data get submitted to a national repository, which in the US is the Biological and Chemical Oceanography Data Management Office, or BICODEMO. And those data are required to be intercompared with other scientists before they're included in the Geotraces intermediate data product, the next one of which is actually coming out in a few days. So as Adrian mentioned, intercalibration is really essential to any kind of program that's going to take place with multiple researchers and multiple platforms. It's also a really great community building activity, and we're going to hear a lot more about that later on. But the way Geotraces handles this is it's method agnostic, so you can use your favorite way of measuring any given element, in this case iron on the left, as long as you get the right answer, and it can be compared with other researchers. Geotraces also employs crossover stations, so each cruise has to cross over a place in the ocean that has already been traveled by another cruise, so that measurements can be compared through time that way. And so these are straight from Bob Anderson about what his takeaway of the lessons learned from Geotraces are. First is to design a program that addresses the problem, so to put the problem first and not the measurements first. To engage agency officers early and often, to establish manageable and achievable goals, to show that the whole program is greater than the sum of its parts, to be patient, that things don't happen quickly, and that early data synthesis provides a basis for future programs. So the example of coming out of the genomics cruises is important as a basis for what might happen next. I also want to say that I personally am a very strong advocate for full depth ocean sections of measuring biology, but I've tried to be more open-minded lately in thinking about what a BioGeoSCAPES program could encompass, and in thinking about what efforts that were not field-based that I think have contributed hugely to understanding marine microbial metabolism. I thought about the Marine Microbial Eukaryotic Transcriptome Sequencing Project, MMETSP. So the researchers involved in this project assembled and annotated 650 eukaryotic transcriptomes. So to me, this is a really excellent example of where a coordinated effort is greater than the sum of its parts, and a study we might think about as we move forward in the next few days. I'm teaching marine microbiology right now and literally just came from giving my midterm. So I also thought I would give this little perspective to some of the early career researchers that it really wasn't that long ago that we were making discoveries with 12 16S rRNA gene clones in the ocean. This is a figure from Giovannoni's first paper on the discovery of SAR11. So there are only 12 clones in this paper. And in less than 30 years, we have from a single expedition, 2,600 near complete bacterial and archaeal metagenomes, which is just truly amazing. And now, using the basis of these programs that I've just talked about, we need to think about how to take this figure and integrate that with geochemistry. All right, that's it. And I think next up is Maite.
Thank you, Alyson. Let me share my screen. Can you see my screen? Yeah? Okay.
That's great.
So first of all, I would like to thank the organizing committee for inviting me to participate. I'm excited to hear your thoughts on this possible program, and I'm really here as a listener more than anything. I would like to also emphasize that there are many individuals involved in promoting the BioGeoSCAPE concept, and this is really an open process, and everyone is welcome to participate and join the effort to conceptualize and bring the program to fruition. As stated here, BioGeoSCAPE is the concept for a potential future interdisciplinary global scale microbial biogeochemistry program. And my talk focuses on the effort taking place to build an international BioGeoSCAPE community. Much of the information presented here can be found in the newsletters in our website, biogeoscape.org. And I put this map here just to show you that there is a lot of interest in BioGeoSCAPE. We get traffic from about 52 countries, which are highlighted in this map. So by the end of all the talks this afternoon, it will be obvious to you that BioGeoSCAPE is a larger effort than anything a single nation could accomplish alone. So currently there is a series of national meetings being organized worldwide. And the hope, as we already heard, is that an international workshop will be organized maybe in 2023 to really design the science plan of BioGeoSCAPE and to come up with a plan to implement it and launch the program eventually. So today, what we have a series of ambassadors of this effort from 25 nations, which are highlighted in the map behind you. We have about 250 newsletter subscribers, and we send a letter twice a year. We have more than 700 followers in our Twitter account. And as I said, we have traffic from 52 countries in the website and 1,500 unique users, more or less. More recently, there was a session organized for Ocean Sciences meeting in Hawaii this coming year, and 44 abstracts were submitted. So in addition to this, we have a series of national workshops that have been completed or are about to be completed. By the end of this year, we will have a total of 7. So the first national workshop was actually organized in Japan by Yoshiko Kondo, Jun Nishioka, and Hajime Obata in September of 2018. In 2019, we had 3 national workshops. The first one was organized by Yaela Shaker in Israel. The second one was organized in Xiamen by Dalin Chi. And the third one was organized by Erin Bertram, Steve Hallam, and myself in Canada. Then the pandemic arrived, but France actually managed to have an online national workshop, just like you are having now, in December of 2020. And this was organized by Catherine Jendal, Ingrid Over-Norsterer, and Damian Cardinal. So the upcoming meetings, well, you have yours right now, which is wonderful. And also France is having their second meeting this December in Toulouse, and I think they're going to try to have an in-person meeting. For 2022, we have 2 big meetings happening in Europe. The first one is going to happen in April in Croatia. This is actually a meeting that was organized by Martha Gledhill and Sandy Orlik, and 15 countries from Europe are going to be participating. And in addition to this, we have another meeting being organized by Al Tagliabue, Thomas Mock, Julie Robidart, and Patricia Sanchez Baracaldo. And this is going to take place in the fall of 2022. And again, it's going to be a pan-European meeting, and probably there will be some international representatives. So I wanted to also bring to you this project that has been funded by the European Union. We are going to hear more about this on Friday from Daniele Iudicono himself, but I just wanted to introduce it to you here. So this is a project that involved 36 partners and for a total of 11 million euros. And in essence, what this is of interest to us because they seek to align their research goals with those of our nascent BioGeoSCAPE program. And in essence, they aim to develop and apply novel and unifying framework to provide knowledge-based resources to better understand and manage the Atlantic Ocean and its ecosystem services, which you can see here. They also hope to develop and disseminate the use of advanced approaches in omics, optics, and sensors. And their cruises will sample microbes, microplastics, and other environmental parameters, including trace metals. And they hope to engage citizen, industry, and policymakers in order to stimulate responsible behavior and blue growth. They also hope to involve experts and pioneers from Europe, South America, and South Africa as well. So more on this on Friday. So in addition to this, there has been a couple of BioGeoSCAPE-like mission cruises. So I will say cruises that have the BioGeoSCAPE sort of a spirit. One was just completed in the Indian Ocean by the National Institute of Oceanography. This was a 90-day cruise and with 30 scientists on board. And in addition to that, we have the Tara Oceans right now is sailing, and they are off the eastern side of South America. And then we have 2 cruises planned by Australian scientists and South African scientists in the Southern Ocean. So what I hope I convey here is that there is a big momentum for BioGeoTraces internationally. And just to summarize, we have 25 national ambassadors so far. We have national workshop or activities happening in 28 nations. And as I said, we have interest on this program by the traffic in our website from 52 countries. So as you have heard, this is really an international effort. So we really need to not only organize ourselves at a national level, but we have to organize us globally. There are 3 proposals right now submitted, one by the European scientists led by Martha Gledhill and 30 others. Also, Max Saito and 4 others submitted one to a CellNet for implementation of this program. And also here in Canada, Erin Bertram, myself, and 10 others are submitting a proposal to do the same. So, and I think getting funded at this level will be very important to launch the program. So in order to move forward, I'm very happy to hear that you're having your national workshop. And the 4 questions that you see here is one of the things we are asking everybody to consider when you have your national workshop. So the first question, here you have the preliminary mission statement we came up with in the 2018 workshop. And we would like your thoughts on this. How could we improve this? In the second point, what we want to hear from you at a national level is how you would like to contribute to the BioGeoSCAPE efforts. So maybe field work or data management or bioinformatics center. The third one is what scientific questions are most important to you. And when you go through this exercise, think about like a 10-year time frame, for example. And then we will also like to hear about any impediments that you might have we want to make sure that you can participate in this effort. And at the end of your workshop, what we would like is for you to submit 2 to 3 page report to the international community in order to share with others. And with that, I would like to finish. Thank you.
All right, thanks, Maite. I think I'm up here. Let's see. All right, that look okay? All right, great. So hello, everyone. Great to have everybody here finally, even if we're online. And my name is Ben Twining, and I'm at the Bigelow Laboratory for Ocean Sciences, and I'm one of the workshop organizers. And my goal sort of with this final introductory talk is to again sort of highlight the motivation for this nascent program that we're trying to build, and then really focus on, talk a little bit about what we've been doing, particularly in the United States towards this goal, following Maite's great sort of summary of the international sort of context, and then talk about this specific meeting and what we're trying to accomplish here. So just to recircle back on this, why BioGeoSCAPES now? And there are, I think, at least 3 really compelling reasons why we should be trying to do this as a community. One, as Adrian mentioned, certainly is our rapidly changing ocean. We know conditions are changing. We already know that nutrient cycles are changing. And we know and have every reason to expect that productivity will be affected as well in pretty dramatic ways and differently across the world. And so I think the science to both understand those and to predict what's going to happen in the future is really critical, not just for the science community, but for our global community. And I think that's why we shouldn't wait any longer. Additionally, as Alyson highlighted, we've made a lot of progress as an oceanographic community in our analytical and computational capabilities that I think allow us now to really take on a program where we try to do a global survey or global understanding of these processes across both omics and geochemical parameters. So I think we can now measure those and have shown that we can do many of those things, including in the organic geochemistry realm. And finally, a really nice sort of stimulus for us to do this is the UN Decade of Ocean Science for Sustainable Development, which you've probably heard about, and the UN has designated 2021 to 2030 to be a decade where, as a global community, we try to focus on ocean science and how we can advance ocean science to benefit the people of our world, in particular in the sustainable management of ocean resources. So just to highlight that, here are 5 of the Ocean Decade challenges that have been identified that I think relate to BioGeoSCAPES. So one of those challenges for us as a community is to understand the effects of multiple stressors on changing ocean ecosystems. And clearly, understanding the physiology and the identity of ocean microbes, I think, is a key part of that. There's a challenge to us as a community to understand and protect marine biodiversity and their ecosystems and fisheries. And here again, microbes make up the base of those food webs and much of the biodiversity in the ocean. And so there again, we need to understand what it is and how it's going to respond to changing stressors. There's a challenge to understand the ocean climate nexus. And this is really key. If we're going to understand how the oceans are going to respond to climate forcing, in particular the biological carbon pump and productivity of the ocean, we need to understand how microbes will respond. There's a challenge to develop a comprehensive digital representation of the ocean. So this is one of the UN Decade challenges. And here again, the idea and the importance of making our data widely available and understood, I think, is a key part of that. And finally, a challenge to enable capacity development and equitable access to data, knowledge, and technology. And this is another really important theme globally that we're trying to weave into the aspects of BioGeoSCAPES to make sure that it's a global effort that benefits everybody on the globe. So, of course, a program like this that we're sort of envisioning has some real challenges too, and things that we need, issues we need to figure out how to, or challenges we need to bridge. And so I've listed a couple of those here for us all to think about and talk about over the next few days. There's the challenge of bridging across scales and disciplines. Just on the scale side, we know these processes go from molecular to global. They go from extremely, short, rapid processes within cells to ones that might take years or decades, for example. And the science spans disciplines, not just within ocean science, but within data science and molecular science and much broader areas that have different communities, different funding sources, and different vocabularies. There's challenges to figuring out the physical and chemical sampling strategies that also match biology. Geotraces came up with one way to do things. And as we tried to figure out how to also make sure we've got the biological processes and the genomics crews, it didn't turn out to be quite as simple as, just adding a smaller pore size filter. We've got certainly standardizing, sampling, and analytical protocol challenges, and we'll talk about intercalibration, particularly on day 2. There are challenges for integrating omics-based technology, particularly applied to the oceans and the environment, I think, with physiological studies and reference genomes that might be created in the laboratory. This is a project that I think will probably not only take place in the field, and so we need to understand how we connect those aspects together. We need to break boundaries between oceanography and omics data sets, so between the communities as well as between the data, and probably develop new mathematical or integrative tools to do that. And finally, again, we need to train a new generation of scientists who can work within and between these communities, a community of omics-enabled oceanographers who are also really cognizant and are thinking about the biogeochemical implications of those omics measurements. So the challenge is ahead of us. So what have we tried to do to sort of start walking down this path? Well, as Maite mentioned, there have been a couple of proposals put in by a couple of national groups. And in the US, a group of us, mostly comprised of the organizers here of the meeting, put together a proposal to the NSF AccelNet program for an implementation proposal. And here's the title of it: Development of an International Network for the Study of Ocean Metabolism and Nutrient Cycles on a Changing Planet, or BioGeoSCAPES. And Mac Sato has been the PI of that. It was submitted last year and got positive reviews, but not funding, and it's been resubmitted a couple months ago. And so I want to share a little bit of the thought process that went into that proposal, which is still pending, fingers crossed. But this gave us an opportunity to start thinking about the steps needed to put this program into place. So one of the things we thought about were the different science domains that need to be brought together for this to work, this program to work. And you might think of these here simply as maybe nucleic acid, all the omics, the sort of disciplines. What we're thinking of is the chemical omics, the things like proteins and metabolites and maybe metals or lipids, things that require a mass spectrometer often to measure. And then sort of the larger biogeochemistry community that measures other aspects that need to be brought in to understand the implications of these omics measurements. So these often are 3 sort of different domains, and we thought about ways that we would need to connect them through a common language of data science, intercalibration, modeling, and also thinking about rate measurements and within these. So AccelNet is often thought of as sort of developing a network of networks. And so we then thought about, well, what are the networks that already exist within these various science domains or communities? And this is a figure which we'll see and you'll talk about in the breakout discussions after these talks. So I don't expect you to read all of these little circles, but these are just examples of the places and the ways that networks already exist within these communities. And the AccelNet project, and I think the challenge for BioGeoSCAPES, is to build connections and bridges between maybe not all of these communities, but members of these communities who can bring their skills to the challenges of BioGeoSCAPES to create this program. And so this is one of the things we really want to make sure we're addressing. And this is a real goal of this AccelNet project, is to connect these networks. So how do we propose to do that? Well, this is a Gantt chart from the proposal, just to give you a flavor of kinds of activities that we're hoping to undertake in the US and internationally to try to bring these communities together. So this includes obviously things as exciting as steering committee meetings happening regularly, but more importantly, these large workshops involving 50, 100 or more people where we talk about science integration, the questions we're trying to address, informatics and data management, intercalibration, and modeling integration. And so we've envisioned large workshops over the next really 5 years here on each of these topics as ways to build this cohesive community around these goals. The SLNet proposal would also fund a significant amount of sort of education and capacity building for early career folks, both through a couple of summer schools that are proposed, professional development workshops for early career researchers, and a number or in funding for early career exchanges for students and postdocs to move between different laboratory groups and institutions, and also mentoring groups that we've thought to create between folks of different career stages where we can work and help mentoring and supporting individual researchers as they work through these different communities. And finally, we put a component in there working with the Ghana Coastal Environmental Summer School as a way to both learn how to and to practice community building and capacity building in developing countries in Africa as well. So another thing we had to think of is, well, how would this be actually organized? And so this is just a draft organizational chart that we pulled together for the proposal to try to give a sense of how this might work. This is very much, sort of a first draft of it. But we've modeled this largely on what we've seen work for the Geotraces program. And so this involves having an international scientific steering committee for BioGeoSCAPES, where members of communities from different countries come together and do that coordination, which enables us to pull this science together and to have a global impact. And that international group likely would have a number of subcommittees that deal with issues such as education and diversity and inclusion, standards and intercalibration, data management and informatics, and knowledge and integration and transfer. So these would be peopled and conducted by members of the international community, including members from the US. We also then envision there to be sort of a US organization that leads the charge on this. So there'd be a US scientific steering committee. And below that, there may be US committees that also try to work on these tasks and activities, basically powered by folks from the US. And these are those sort of activities that we proposed in the proposal. And I want to stress that first of all, that this organizational chart is very much a sort of an idea for discussion and proposal, but also that there are no names associated with these yet. This is very much, an opportunity for folks from throughout our community, nationally and internationally, to get involved and move the ball forward on these various topics. Sort of towards that end, what we envision is that those of us who have been involved in developing this proposal would not necessarily form those committees, but rather the OCB Scientific Steering Committee would sort of stand up the BioGeoSCAPES Scientific Steering Committee through nominations nationally. So that would be outside of the group that has sort of worked to pull these together so far to ensure inclusivity and a really broad and representative sort of leadership on this potential program. Okay, so it was exciting to look at organizational charts, but why do we bother with an organizational chart? And I want to finish here just by highlighting some of the hoped outcomes for such a program, because hopefully these excite us and that there's many others that folks here can think of to add to this. But one thing that we hope that will come out of this really is a baseline understanding of microbial communities and their metabolic function in the ocean. And arguably, we might understand this for certain areas or certain times of the ocean, but we certainly don't have a global understanding of this, and that would be a goal. We hope that there will be new tools for data integration, visualization, and analysis that will be developed. We hope to quantify biological and biogeochemical hierarchies that structure ocean metabolism at different scales, thinking about the emergent properties of these ecosystems that have biogeochemical implications for our climate. We hope to understand the key biological feedbacks that should be represented in ocean ecosystem models in order to really let them predict what will happen in the future with our ocean. And we want to lay the foundation for the study of future anthropogenic impacts on the ocean and the ocean's resilience to that. I think those will all require a more mechanistic understanding of the microbes at the base of these cycles. And finally, our goal is to train a new generation of scientists worldwide for increased capacity in omics, bioinformatics, modeling, and biogeochemistry. Okay, so that's the big picture. Where does this meeting sit in that goal? Well, we have hopefully some pretty discrete, straightforward goals for this meeting. One is to communicate the current status of the international and US BioGeoSCAPES planning effort. That's really been the goal of these first 4 talks, to get everybody up to speed on sort of what we've done so far. We hope to engage US-based scientists from a range of disciplines who can help to develop and champion this program. And that is the goal of the next 3 days, is to get everybody here involved in thinking about what's possible and how we're going to do it. And finally, we want to identify the US science priorities, or we start to identify them in ways that we can contribute to both a US and an international science. As Maite pointed out, that's one of the things that the international organizers are asking for, is each country to think about what matters in particular to us and how we can contribute. So with that, I think that's the end of our introductory talks. And I think we have about 15 minutes or so for questions and sort of initial discussion. Thank you.
Great. Thanks, Ben, and all the speakers. My name is Max Saito. I'm also one of the organizers, and Naomi and I will be emceeing today's session and facilitating this Q&A session. So feel free to put your questions in the chat, and Naomi and I will alternate picking and trying to answer or pass them on to people to answer. Let's see, why don't we start with, I see there's a question from Drew Chappell. Are there any key steps in Geotraces planning process that we are skipping? I didn't see the intercalibration cruise in the timeline. When did that happen? And in the timeline for getting Geotraces off the ground. That was a great point. I can add briefly, I think the cruises were in 08 and 09, and there was a safe cruise as well. Before that, that was an iron intercalibration cruise. There'll be more conversation tomorrow about the intercalibration efforts and just how important that is, and the challenges, particularly in big data omics space, and a lot of new parameters that people may want to try and think about studying in the BioGeoSCAPES concept. Alyson, did you want to weigh in on that question as well, or anyone else?
No, I think you hit it, and Kristen also as well hit it in her response in the chat. I did put a link in the chat to all the presentations from the Omics Intercalibration Workshop are public on an OCB website. So if you want to check those out and see where things are going. But yeah, we're going to hear more about that tomorrow.
Great.
And sorry, I left it off the timeline. That is an important thing to have left off, but maybe we can blame Bob Anderson.
Let's see, Naomi, I'll pass it off to you.
Yeah, so there was a question from Anon. I don't know if you want to very briefly phrase it, Anon, and then we can start a discussion there.
Sure. One of the things that has come up throughout the sort of ocean chemistry and biology discussions, it happened in JGOFs, it happened, I think, at OCB, is where do you cut off? Right? Do you cut off at the phytoplankton? Do you cut off at the zooplankton? Do you cut off at the bacteria? And I wonder if there's been thought about ways of incorporating, maybe through eDNA, the whole ecosystem, right? Especially given that there's increasing, at least in coastal ecosystems, there's increasing evidence that maybe some of the important bacteria are not actually out in the water column, but they're in the guts of organisms.
Yeah, that's a really great question, and it's something that we've talked about as organizers and as something that we feel the community needs to think about. And it's a really hard question of how to be inclusive and capture all the really important processes, while at the same time not being too broad that it's not clear what a BioGeoSCAPES program is actually aiming to answer. So I'd love to hear other people's thoughts, but just one thing I wanted to throw out there is while we have kind of targeted topics for the breakout rooms over the next couple of days, there are spaces, and I'll show you where they are, in GatherTown, where we'd really encourage participants to self-organize. And if there's a group on the workshop that wants to kind of get together during the poster session or after the poster session, GatherTown will be open. And you could have this discussion and create a Jamboard slide that will be kind of incorporated into the workshop report. So the Jamboard slides that we'll show you for the breakout rooms, again, we want to try to keep the breakout discussions, they're pretty short, targeted on the questions that we're asking each day, but there is plenty of room, and really would welcome these extra perspectives and kind of discussions to happen, and we can help facilitate those questions. Any other questions on that, comments on that? Great. Matt, do you want to?
Sure, I can jump in. I think, yeah, I think that's a really exciting and interesting question, and I think that's one that, you know, this is very much a bottom-up effort, right? And at the community, you know, for all things that could be studied in BioGeoSCAPES, I think the community needs to try and self-organize and try and figure out what's feasible and tractable. Let's see, Jim, I see you have a couple questions. Maybe let's start with your second one. Do you want me to read it, or would you like to say it?
Oh, sure. Well, before Geotraces actually was sort of announced to the community, there had been a lot of interest in intercalibration. So people within the chemical oceanography community making common measurements. And prior to that, most of that kind of work had been done by the DIC folks, you know, like led by Frank Malero and others, you know, how everybody could be on the same page measuring pH and alkalinity and so forth. And so when Don Rice wanted to support intercalibration efforts for things like iron, his first stop was having a group of people participate in an NRC report, which had basically a bunch of people involved with making measurements get together and discuss priorities, getting involved with people from organizations like NIST, who are involved with reference material and protocols and things like that, and come up with a report. And these are pretty standard. Probably some of you have been on these things before. They're hosted by the National Academy, and there's usually a one big meeting where they bring a lot of folks together as well to talk about priorities. I don't know if it's relevant here or not, or if there's something like that already been done, but this was an exceptionally valuable document to point to in terms of getting money for things like intercomparison efforts and things like that. And so I was wondering if there was any, you know, if the organizers had thought about approaching the NRC about sponsoring one of these things.
Great, thanks, Jim. I can start this and then pass it off to some people. But I think that's a great idea. And you know, how do we begin these intercalibration efforts? You know, there's a bit of a culture shift. I think the chemists maybe are a little bit more entrained in intercalibration, and I think the biological community is learning and excited to try and delve into that as well. We do have a participant at this meeting who will be from NIST, so he told me that he would be here a little bit later, so we will have some representation there. And the efforts thus far have largely been through OCB's support, the workshops and the metaproteomics effort that I'll talk more about tomorrow. Let's see, maybe I should pass this over to Adrian if he has anything to add to it.
Yeah, I mean, I think, you know, again, similarly, there'll be a presentation by Paul about the outcomes of our nucleic acid omics standardization intercalibration workshop. What came out of the workshop was definitely that there's some work to do at all levels, as I was mentioning, from sample collection to everything to you know, standards that we would put into the samples, including external standards, internal standards, mock communities. And so the work that we've outlined that is needed would likely need some sort of funded effort to accomplish. And so we're in the process of hopefully achieving that. But I mean, I think Paul will also be giving an update on those efforts tomorrow as well.
Just a related question. Is there any plans for something analogous to the Geotraces cookbook coming out of your, I mean, I realize that could be a minefield with some of these techniques, but I just
Yeah,
wondered
and
if it was in the mix.
absolutely. I mean, and those efforts are actually underway, even with, one of the interests in Atlantico, which is, again, a funded program, is coordinating, all of these sort of sampling efforts and intercalibration. And, they've actually even gone forth and started putting together best practices and things like that. So, I mean, again, it's a bit of a moving target for certain parts of it, but I think that, we've come to a point now where we can come to some sort of consensus on certain parts of the pipeline, at least for the nucleic acid omics.
Thanks, Adrian. I think, Alyson, did you have something to add to that?
Yeah, I just wanted to follow up on Jim's comment about, first, the suggestion about an NRC workshop is really interesting, but I also wanted to mention that the human microbiome community does have reference materials, so it's definitely not out of the realm at all of things that some microbial scientists are thinking about. So they made a big push to try and develop a standard reference material for human gut studies. And that's a model we could turn to, thinking about what happens next for this community.
Thanks, Alyson. Amy, should I pass it back to you?
Sure. All right, so I'm gonna surprisingly pick out Heather's question about models. Heather, do you want to ask it?
Sure. Can you hear me? Sorry, I'm in a coffee shop. I'm Heather Kim at Hui. Yeah, I was just wondering if you guys have a working group for comparison of all this different omics and biogeochemical modeling, just like CIMIP-6 or Merriman, which is the side project of CIMIP in the past. It'll be good to talk about in terms of data science aspects and how to calibrate and compare all these different models and modules of omics and biogeochemistry.
So yeah, absolutely. I totally agree with you. But so there isn't one currently, but this is something not just related to models, but these are kind of bottom up groups that have organized. And so the other intercalibrations have been other groups that have come together, like the proteomics group that says we really need to do this intercalibration. And I agree with you. I think as modelers, we need to do that work as well. And it would be fantastic to organize and put together workshops. So yeah, I hope that maybe at some point the modelers that are on this call we can get together and maybe put in a proposal, because I agree that would be really vital, but hasn't happened. So yeah, there's not a kind of pre-plan about what needs to be done in terms of intercalibration, and that's one of the things we hope will come out of the discussion tomorrow, is identifying this need, and then hopefully people will self-organize and step in and help fill that need. I don't know if any of the other organizers want to chime in.
Yeah, I think one thing to point out also is that the AccelNet is really a proposal that NSF does to try and help forge international connections. It's a really interesting concept proposal. It doesn't really fund science, it really forges networking, and networks of networks has been mentioned. And so we recognize that modeling is a really crucial part of developing this program concept. And so I think there's a whole workshop effort within that proposed activity. Let's see, why don't I move on to Julie? She's got a question here. Would you like to ask your question, Julie?
Yeah, I'm just sort of curious about money and scale here, and sort of the scope of what you guys are envisioning and how it compares to some of these other big programs that NSF has sustained. Just to, again, as a deep sea person, right, who uses a lot of infrastructure, I'm curious what you guys have talked about for this program. Is it kind of like the Geotraces model, where you're using existing infrastructure, which definitely saves that program a lot of money, I think, but, or, do you want a dedicated ship that, and that kind of stuff. So more just trying to get a sense of your vision.
Thanks, Julie. Maybe I'll try and start this answer and others can chime in. I mean, I think, as we said at the very beginning, this is the concept of BioGeoSCAPES is larger than any one country can really try and do, right? And so the hope is by pooling resources and ship time scientific, minds really, can it really push the field forward? And I mean, Geotraces was kind of an exciting example of that, where each country contributed what they could in terms of ship resources, and other countries were able to participate in those ships. Interestingly, though, Geotraces itself was funded in the U.S. out of NSF Chemical Oceanography Core program, right? There wasn't a dedicated call, and I think Mike Saraki and may talk about this more on Friday. So it was within the scope of that program. There's some hope, I think, that resources could be shared or developed that are in Geotraces or elsewhere, at least in the U.S. But I think I think we don't have dollar amounts yet. This is very early in the program of conceptualizing in a bottom-up effort to try and convince that not only NSF but other nations that the community is behind this and wants to see this go forward. That said, though, I've heard Bob Anderson say that Geotraces has collectively globally has hundreds of millions of dollars, if not more, that's been invested in it. Anyone want to add to that?
Yeah, Mac, I'll just jump in. I mean, I totally agree with your answer. And I think, Julie, I guess I hope that what we come out of is an idea for science that we can then go and advocate for those funds. I mean, definitely NSF has not committed anything to this, but as Mac said, reading the interest of the community and seeing what size community can we motivate and get behind this idea, I think will drive resources in the future. I'm guessing that Bob hasn't said it to me, but I'm guessing that the Geotraces didn't necessarily start with, what could they didn't have a dollar value in mind that they start with, but I think as they demonstrated early successes, both nationally and internationally, I think the resources have lined up behind it. And I guess that's maybe my vision of how this will happen as well, is that if we can get some of these first steps funded, whether it's the AccelNet or intercalibration exercises or some projects that can demonstrate the value of it, or that we can look at existing projects that do that we can use that to go advocate for those resources, whether they come from core or whether they're new monies. Yeah, that's, I think, how we're approaching it.
Yeah, I would just say as an outsider, I think you guys have already done that. Just there's so many of these studies in the water column. I think you have already done it. And the only reason I bring up these programs is because, for example, OOI, there was a huge groundswell and then this giant investment. Like, it happened very quickly. It's just something that I don't know, should keep in mind. That's all.
Yeah.
Something else that's worth bringing up is that there's been some discussion about maybe BioGeoSCAPES could have a slightly different model than Geotraces, that instead of having one cruise every 2 years, perhaps it could be more of a franchise model where there could be science goals and questions, and communities could self-organize and have some smaller projects and more nimble projects that could then be more frequent and get a lot more data that we may need to cover the biological and temporal variability that's out there. So that's something to keep in the back of your mind too, is how could something like this not only leverage more science, but also involve more people along the way too.
So I think for that, we're going to switch gears just to keep us on track. I'm sorry if we didn't get to your question in the chat. Before we introduce the breakout discussion topics, we're going to do a workshop picture, which will be a screenshot of the Zoom, unfortunately. So if you're able to turn on your camera, that would be great, just for a couple minutes or 2 seconds, I guess. May is going to take some screenshots for us, and then we'll introduce the breakout today. All right.
We look good.
And
you all look amazing. I'm almost done. So engaged.
Try not to blink.
Okay, thank you. I've got everyone.
all right.
Great. Let me share my slides. So we'll transition to the breakout now. Can you see my slides yet?
Yep. There.
Yep.
Great. So I want to say, we're all really excited that we had such a great registration turnout, and we have over 100 people that have already logged on today. I know people have to come and go for various obligations, but we're super excited about this, and we really want to take this opportunity to maximize input. to hear from as many voices and perspectives as possible. There's a lot of people here, so we want to get that feedback in, and so we want to use these breakout groups to have to facilitate multiple discussions in parallel and bring that information back. Oop, let me get my slide to go forward. There it goes. And so to do this, we're going to use Jamboards to try and collect the information. And the idea is that this will help us capture key discussion points. If people want to keep text-based notes as well, you're welcome to do so and email them to us. But the advantage of the Jamboards is that everyone can see each other's Jamboards afterwards. And so that will be some of our homework to go back and tonight and look over what other groups talked about and sort of think about, incorporate that thinking into your conception of this. And importantly, we will use these Jamboards and the notes in the workshop writing report, which will become the basis for our US perspective on this. And so how this works is the Jamboards, basically, there's a link provided, I think, in the email and the chat as well. But if you can go to page 7, there'll be the day 1 sessions, and you go to the page that's labeled with your breakout room number. So we'll do automatic random breakout rooms, and that number that the room in will then correspond to your Jamboard. Add your participant names and capture discussions either with a note taker, or everyone can put their sticky notes up however you see appropriate. But please create a summary sticky with key points so that we can people can read it and get a summary sense. And one thing we want to try and propose to try and facilitate this is using a circle process. A circle is a shape where everyone can hear and see each other, and it's a group process that supports every voice in a collaborative decision making. And a circle is an experience of sharing a story and inviting diverse thinking and encouraging creative problem solving. So since we have such a large group here, we would like to suggest that everyone start with the circle process, begin with the breakout room, with each member of the group providing their initial thoughts. I know you don't have a circle in Zoom, but each person could pick the next speaker so that everyone gets a chance to speak up. That way we get at least a chance to hear from everybody. And if people want to suggest halfway through that maybe we should do this again or something, please feel free to do that. And I want to revisit our code of conduct as well. Give everyone an opportunity to speak. Don't interrupt this person speaking as the floor. Speak with intention, noting what the relevance is to the conversation, and be respectful. Critique ideas, not individuals, and listen with attention, and tend to the well-being of the group, remaining aware of the impact of our contributions. So with that, our day 1 discussion topics are what are some examples of successful community building efforts, and what are the lessons learned? Again, thinking back to that cloud of networks, how can we bring people in, and what are we missing? What are the potential barriers to interdisciplinary and inclusivity in a global effort like this, and how can we address them? And what are the key communities that should be integrated into BioGeoSCAPES? So with that,